Thank you very much, Tina, for the introduction, and also Chad and Anais for having me here. I have to say that the structural bioinformatics is my interest, but it's not the interest of my group anymore, which means that we're not doing this. Only one people and myself. So, can I start now, or shall I wait? I can wait. Do anybody happen to have a pointer?
I think you can use it on the.
Yeah, there is an option.
No, and
Here, this one, and then here we can use this pointer, and then you can just record this.
So yeah, whenever you give me green light.
yeah, I think you can. Maybe we can have a technical issue just to say. Like randomly click on something on the computer.
I can add 50 extra slides or something. Okay, very good. Let's start. So basically, I guess that everybody knows what a complex system is, but just in case, so the definition is a complex system is that where the whole is more than the sum of its parts. And this is very trendy, but I'd like to remind you. So this is, for instance, this is a social network, and is very well known that it's difficult to predict the global effect of a social network by studying each one of the individuals that are connected to this social network. So we're doing biology. So the same happens here, right? So if you study individually the movement of each one of these sardines, it's almost impossible to guess what is happening. And what is happening is that there's a diver that is interfering with the whole school of fish. So by studying each one of these elements independently, we cannot just grasp what's the global effect that we see. And of course, human biology is a very, very complex system. So this slide is mainly for the engineers in the room. So we have something like 23,000 unique genes, about 100K unique proteins, 200K unique metabolites, but we have something like 40 million proteins per cell, and we have something like 30 trillion human cells. So this is for the people that are impressed with this 1.5 or 1.7 trillion parameters in GPT-4. So human is a lot more complex, and as a friend of mine says, and it works with only a bowl of rice per day. So this is the energy that we use, right? So I'm at the Institute for Research in Biomedicine. It means that on the one hand, we aim at understanding disease, at understanding human biology, but then on the other, we try to look for means to fix the pathological problems that we might encounter, right? So this, we see it as an equation between the disease biology and then the chemistry, which, I mean, chemicals are still the best assets that we have to treat diseases. So basically for very many, many years, so what we were doing was purifying these active compounds from nature. So we were not synthesizing anything. So this came late in the 18th century. So what we had is that somebody saw that by infusing the white willow tree leaves, the headache or the pain would go away, or of course by chewing some coca leaves, your dizziness might go away, right? Then at the end, what we did is say, okay, so there must be something in this white willow that makes the pain go away. And then what we did was to purify these compounds. But then in the 18th century, we started playing chemistry. And this means that we started synthesizing these compounds, not just purifying them. And something like 70 years ago or 80 years ago, we started playing also with molecular and cell biology. This means that it was a very, very good means for simplifying the complexity of the human body. So this was great because we didn't have to work with the whole human body. We could work with one individual protein or with one individual cell much easier, right? So this means that if we could play with synthetic chemistry and we could play with individual proteins, we could go work in the test tube and synthesize molecules that would inhibit, for instance, the function of a particular protein. The problem is that every time that we cross one of these that we call complexity barriers, we gain understanding, but we lose information, right? So then if we work at this level, as I said, at the test tube level, where we have our purified protein, our compound, then this might not work in cell-based assays. This might not work in animal models. And finally, it might not work. It almost certainly will not work when we transfer it to the whole human complexity. And this is exactly what we know as the drug discovery funnel. So you can start studying thousands of compounds, then you can do experiments with a fewer thousands of these compounds that work in cells, in animal models. But when you go to the clinics, only one of these thousands of compounds, if you're lucky, will work. And it's not that we have millions of drugs. I mean, we have very few drugs approved. So roughly 6,000, 7,000 different drugs. And we're able, the whole world is able to discover something like between 30 and 50 new compounds per year. So very, very few. And one of these problems is this complexity, right? So removing biological complexity from the drug discovery pipeline. So what my lab is trying to do is to reintroduce, as much as we can, this biological complexity from the first stages of the drug discovery pipeline. And of course, this is 2024. So how are we trying to do that? By merging and integrating information, data, and artificial intelligence or machine learning. So this is the network biology session. So we're using networks here. I mean, as Tina said, my background is on network biology, structural bioinformatics. But here, I mean, we can use networks in a different way. So of course, we all know that what is a node, an age, and we're used to this type of representation. So every node is a protein, and two proteins are linked if they do physically interact. We can go a little bit more complex and represent pathways or represent complexes, but this is basically is the same idea. So the nodes are physical proteins that they do interact. But we are also used to use networks as a way of representing, a very powerful way of representing relationships between biological entities. So when we have this gene disease network, it doesn't mean that these genes do physically interact with a disease. It means that they are associated to a disease, right? And then we can use this to study biology. But of course, as I said, biology is very complex with very different players. So we not only have proteins or genes, I mean, we have cells, we have tissues, we have different diseases, we have compounds, etc. So we need a more complex network. And this more complex network is, in one case, what we call knowledge graphs. So we're not using multi-layer networks like we have seen several talks. We're using these knowledge graphs. This one was introduced. It was the precursor of the spoke model that Sergio introduced yesterday. It's called the EtioNet. It came out from his group in 2017. And basically, this knowledge graph is just an heterogeneous network where nodes can be of different types, and the relationship between these nodes can also be of different types. This is what we know as a knowledge graph. Networks are beautiful and are easy to understand the concepts, but they're a pain in the neck to work computationally, because most of these relationships are very sparse, and then when we transform this into a matrix, we get something that is almost unbearable from a computational perspective. So what people are doing now is using what is known as network embeddings. So it's basically reducing the dimensionality of these huge and sparse matrices into some vectors, some descriptors that are a lot more compact. And if we're doing our job well, this network embedding, if it's well done, so we should be able to preserve both the local properties of these networks, but also the global properties. So we can preserve the idea that these two nodes are very close in here, but that these two groups are far away. And this is exactly the trick that we're using in the laboratory. We're playing with knowledge graph, networks, and network embedding techniques. As I said, we're dealing with an equation that has chemistry on the one hand and biology, disease biology on the other. So let's see how we can format chemistry first. So for 30 years, chemoinformaticists have been exactly encoding the structure of small molecules, right? So if you see this molecule, unless you know exactly what it is, you cannot get its function. But we have very many different means of encoding this in the form of a vector, usually, and this is something that the computers understand, and we can use this to compare different compounds. And what we know is that whenever two different compounds have similar chemical structures, they used to have similar functions, right? So we can do it to the fingerprints, the structural keys, scaffolds, etc. Then what Miquel Duran set up to do in my lab already 7 years ago, saying, okay, so can we encode not the chemical structure of these small molecules, of these compounds, but their bioactivity effects, so the effects that these small molecules will have when we expose them into a biological system. And this is exactly what he had to do. And the first step in here was to collect experimental information. So we created something that we call the chemical checker for very obvious reasons, and is basically a resource that is based on experimental information for over 1 million chemical compounds. And we have divided this chemical checker into different levels of complexity, following a little bit the way we understand drug action. So at the most basic level, we have the chemistry. So this is what we've been doing for 30 years. We have different, what we call bioactivity spaces, 2D fingerprints, 3D fingerprints, the scaffolds, etc. Then obviously for a compound to be bioactive, it means that at some point it has to bind a biomolecule. So this is our second level of complexity, the targets. And then here we have descriptors that represent the mechanism of action, the genes that metabolize these small molecules, binding, etc. Then when you modulate the activity of one target, this modulation gets spread out through the cell through different types of networks. And then in here we have signaling pathways, small molecule pathways, interactome networks, etc. And all these are effects that we can capture in cell-based assays. This is our fourth level of complexity. So we have heard about the LINX 1000 a couple of talks ago. So this is what we have in here. So gene expression. So you have your favorite cell panel. You put the different compounds. You measure how the transcription changes in these different cells. Or we have the cancer cell lines. We have also heard about the GDSC or the different panels that we have in here. We have cell painting assays. You put the compound in a cell, look under the microscope, measure 900 different features of the cells, etc. And finally, the highest level of complexity is the clinic. So for those few compounds that reach the clinics, we have descriptors here that tell us about the therapeutic areas, the indications, side effects, etc. So I'm not going to go into details because the chemical checker and all these descriptors have been around for a few years already. But basically, to cut a long story short, what we have now is 25 different ways of describing the small molecule bioactivity, right? So we have these descriptors, vector-like descriptors, 128 dimensions, that basically tell us not only about the chemical structure of these compounds, but about the likely targets that they're going to bind, the network perturbations, the effects on cell-based assays, and finally, the clinical effects. So we have a way of describing bioactivity in a format that the computers understand. This for the chemistry. How about the biology? So the problem here is a lot different because we have a squillion different chemical molecules that are very poorly annotated. In biology, we have a few players that are hugely annotated, but we also have to take care of that. So basically, as I said, so we have genes, proteins, compounds, cells, tissues, etc. And there are very many different ways of encoding these different biological entities. We couldn't afford to play with a squillion different types of encoding, all these properties. So again, we reverted to the knowledge graphs, and Adrià, a very talented PhD student in the lab, kind of evolved the Etionet that Sergio presented yesterday in something that we call the biotech. Right? And the biotech is a gigantic knowledge graph, biological knowledge graph, that revolves around 12 different types of biological entities. So we have genes or proteins, we have cells, we have tissues, we have diseases, etc. We populate this knowledge graph from information coming from over 150 different databases. And what we define here is almost 70 different types of relationships between these nodes. So we have that the gene might be upregulated in a cell or related to a disease or targeted by a compound. So overall, the biotech contains information around these 12 different biological entities, more than 500,000 nodes, and more than 30 million different ages. So, and what we want to do in here is, as we did for the chemical checker, try to encode these information in the form of a vector. So what's the process that we used here? So I'm a biologist, I'm a biochemist, So we didn't want to lose completely the control of these embeddings, of these descriptors. It means that we didn't embed directly the knowledge graph. So we identified different types of what we call metapath. So different ways of traveling through this huge knowledge graph. For instance, we want to find descriptors that tell us about relationships between compounds and diseases going through their target proteins. Nothing else. So only these types of relationships. Then what we do is we go to the biotech, we travel through all these different types of nodes using only this metapath, right? And then basically we transform this into the corpus of information that we then embed again in the form of vectors. For connecting them to the chemical checker, we use 120 dimensions in here. And basically what we end up is for this particular metapath with 3 different types of descriptors. So something that tell us about the compound, something that tell us about the diseases, and something that tell us about the connections between compounds and diseases that take into account all the information that we have in here. Of course, the first thing that we have to do is to make sure that technically the embeddings are correct. So these embeddings, these descriptors are able to recapitulate the original structure of the network, and then seeing whether they are really able to capture the biological effects that we want. So overall, we ended up in here not with 25 types of descriptors, but with over 1,000 different types of descriptors that we call context dependent. Why is context dependent? Imagine a protein. So we can describe a protein according to its name, to its amino acid sequence, but also according to their interaction partners, right? Or seeing whether it's expressed in certain cells or not others, or whether it's related with diseases or not, present in certain tissues. So all these are these 1,000 context-dependent biological descriptors that we have in here. And as in the case of the chemical checker, that I didn't go into details, so this information, the most important thing is that in some cases it's redundant, but in some other cases it's orthogonal. So imagine 2 genes in here, for instance. 2 genes that have very similar function. So I don't know if you can see here, but you trust me that their descriptors in the functional space are very similar. They also share subcellular localization, so very similar in here, similar biological processes, but one of these genes is expressed in a tissue and the other gene is expressed in a different tissue. So these 3 descriptors are going to be almost identical, but this one in here is going to be different, right? If we go at cell lines, for instance, 2 different cell lines that harbor the same genetic mutations, so the mutational space, very similar descriptors, also in terms of gene expression, but when we go at the protein level, we see different abundances in here. So this means that there's differences in the clearance, similar expression, but dissimilar protein abundance. And of course, this translates into different drug sensitivities. So in this case, we have 2 descriptors that are very similar, but 2 that are completely different. Yep.
Are these all in the same space now?
These are all in the same space now. What do you mean? We are providing.
Like are the embedding spaces?
No, we have
to each other.
We have the over 1,000 different embeddings in here. They're not connected. So, and then what we could do, and this kind, I mean, we want to think that all these embeddings sort of capture most of the known high throughput biology, at least. So we want to use that to assess the support or the novelty of new databases, right? So for instance, we collaborated with Mark Vidal and others on the human reference interactome. We charted over 53,000 different protein-protein interactions, and we want to see how many of these are novel or how many of these are supported. So basically what we can do is we take all these pairs that we discovered in the lab, we go to the different embedding spaces that we have in the biotech, and then we just compute distances between the descriptors of these 2 proteins in each one of the spaces, right? And then basically what we see in here is that there's a few of these 50,000 proteins that are known and supported using other types of data. some that are known. This is basically from previous experiments that we did, Curie 2, Curie 1, etc. This is Curie 3. And then there's something like 1/3 of these interactions that are completely novel, but are supported by other types of experiments in here. And we have finally something like half of 40% of the interactions that are completely novel, right? So this allows you to assess the novelty of any data set that you produce. And since this was pretty useful, so we transformed this into a server that we call biotech supports, where basically you can input any set of relationships between biological entities. It could be cells, it could be proteins, it could be genes, mutations, etc. And then it looks for support. So this runs automatically, and basically what you get is exploring these 1,000 different context-dependent descriptors that we have in here. We evaluate which ones support your data, right? We can also evaluate whether the support is higher than expected. So you want to support your data, but you want it to be novel as well. And this is what Chad asked yesterday. So we're also providing information as to which ones of these metapaths, which ones of this information contain data that is related or are more informative to predict new interactions in this case, or new relationships of the same type. So you have which ones are the most supported pairs, but also which ones are the type of metapath that really support this data. And if you want to implement any type of predictor, the ones that you should use. Good. So now we have, again, to recap a little bit, we have vector-like descriptors for small molecule bioactivity, and we have vector-like descriptors for biology. So what we want to do now, of course, is to merge these two different worlds. And basically, this is what we call a general connectivity. So in here, you have your favorite biological descriptor, either coming from the biotech or that you come up with in the lab, and you have small molecules, and we can connect them. So we've been doing this for many years at the target level. So you have your target related with a certain disease protein, and then the compound, and you were expecting that this would serve as as a good medicine for that or a treatment for that disease. But now we can go higher up in this complexity scale. So we can look for compounds that not only modulate the activity of one protein, but also the activity of a network, or the activity of a whole cell, or even compounds that are able to treat something as abstract as disease symptoms, right? And this is by just doing mathematical operations and looking for chemical descriptors able to match or to revert biological descriptors. Examples on that. So you know that is very trendy and is a powerful tool now that we have biological drugs, so monoclonal antibodies, etc. So this is good because they're very specific, but they're very difficult to produce, difficult to handle, and very expensive. So if we can find small molecules that do exactly the same effect than these biologicals, this would be a good thing for us and for the pharma industry. So this is just one example of several that we did. So in this case, we're trying to mimic the effect of daclizumab. So this is a drug that is currently being used to avoid rejection upon organ transplantation. And what it does is it blocks the interleukin-2 receptor, right? So the experiment that we did in here, we went to the same cells that these guys used to discover or to come up with daclizumab, PBMCs. We genetically killed the IL-2 receptor, and then we ran some differential gene expression experiments between the cells with the intact receptor, the cells with the killed receptor. And what we want to do here is to find a small molecule that is able to mimic this genetic blockage, right? But what is important is that we're using gene expression data, but we don't care about blocking directly the interleukin-2 receptor. So that's why we are also introducing 3 types of network descriptors in here. So we want to mimic the global activity of the network. We don't care about the specific protein that this small molecule or daclizumab are targeting. So what we did is basically mimic the same experiments. Well, we went to the checker first, of course, and then we identified 19 compounds that we thought could do the same effect. And when we tested them, on cells, we found that indeed 14 out of these 19 were very clearly able to block cell proliferation dependent on interleukin-2. Some of them were very potent, right? So this is just one example. This is an uncharacterized compound. So this is the experiment that we did. So you have cells here, untreated and without interleukin-2. They do not proliferate. When you add interleukin-2, but no drug, these cells start proliferating in here. And when you add daclizumab, this monoclonal antibody, of course, you stop the proliferation of these cells, right? So when you add our compound, uncharacterized compound number 20 in this case, we're doing the same and in a very nice dose-dependent manner, right? So we are able to block completely, even more potently than the daclizumab, the proliferation depending on interleukin-2. What is nice is that we know that downstream of interleukin-2 receptor is the p-STAT5 that gets phosphorylated upon binding, right? And this is what we see in here. So we see that when there's no drug, this STAT5 starts being phosphorylated. When we add daclizumab, phosphorylation completely stops. With our drug, this phosphorylation of STAT5 is completely unaltered, which means that whatever we're doing, we're achieving the same effect, but through a different target, right? So this is a result of using these network descriptors in here. Other examples. So of course, this is experimental, but we need to cross-validate our methods like we do in computational biology in here, right? So what we did is to come up with a new cell type that the chemical checker has never seen. So in this case, we were studying Alzheimer's disease, and we were using these SHSY5Y cells, kind of neuron-like. So what we did in the lab is to introduce mutations that are related to Alzheimer's disease, right? So this is completely new. We don't have information on Alzheimer's. We don't have information on these cell types or these mutations in the chemical checker. And then again, we run differential gene expression experiments between, let's say, the unaltered or the native neuron-like cells and the neuron-like cells with Alzheimer's disease mutations. And in this case, what we want to do is to revert, to find small molecules that are able to revert the effects that these Alzheimer's disease mutations cause in these cells. Again, we went to the chemical checker and we found a bunch of compounds that computationally were able to do that. We tested them experimentally in the cells in the lab, and we found that indeed, to a certain extent, we are roughly able to revert 50% of the changes that these different mutations induce in here. And these are not random changes. So basically, we're reverting the effects on very well-known genes that are related to Alzheimer's disease. So BIN1 is implicated in vesicle endocytosis in the synaptic membranes, or we have the glutamate receptor, etc. Right? So something that comes out of the computer works in cells. But what we claim in here is that we are reintroducing biological complexity at the first steps of drug discovery. So cells is not enough. So what we did is we went to mice models, in this case, 3 different models for rodent models for AD. And since the disease starts in the hippocampi, so what we did is to study the hippocampi of these animals, of these different models. We did full transcriptomics, full proteomics of the hippocampi in here. We did some standard bioinformatics, identifying changes, etc., wild-type mice versus AD mice in the different models, different time points, etc. And to cut a long story short, what we identified is this module, this group of proteins that is completely unaltered. I don't know if you guys can read it here. I certainly cannot. But basically, this is aging, right? So this module 2 is completely unaltered in healthy aging, but is shooting up in Alzheimer's disease models. And this happens both at the transcriptional level, but also at the protein level. So this is the module that we want to revert the effect. So we played the same trick that we did for cells, in this case for mice. Of course, this is mice, which means that we cannot use any funny compound that we have in the laboratory. So we have to use approved drugs in this case. And basically, what we wanted to check is whether we're really hitting this complexity. So we went first to phenotypic assays with these mice, right? So in this case, we let the mice grow until they were 5 months, and we treated them for a month. And basically what we checked was the cognition capacity of these different mice using an assay that is called novel object recognition. I don't know if you're familiar with it, but basically you have a box with two identical objects, and then the mice explore them, get to know them, etc. And then after a week, you replace one of these objects by something that is new, and mice are curious by nature. So the healthy mice, what they do is they spend more time exploring the new object than the old one. The AD mice, since they don't remember that they know this object, they roughly explore 50% of the time the same object. And this is exactly what you see here. So for the AD mice, they spend 50% of the time on each object. For the healthy mice, they spend roughly 60% on the novel object. So after we treated these mice with the 6 compounds that we identified, we could see that for 4 out of these 6, we could indeed, at the phenotypic level, revert these effects, right, to the level of completely healthy mice. So fine at the phenotypic level, but is it working because of the reasons that we say it's working? So then after the phenotype, we had to go again at the molecular level, and what we saw is that indeed these 4 compounds were able to downregulate the genes that were overexpressed in AD mice, and only one of them, that is the one that had the highest effect also at the phenotypic level, was also able to upregulate the genes that were downregulated in the AD mice. So it seems that also at this mice level, we're kind of hitting this complexity with very good numbers, because we're saying that 4 out of the 6 drugs that we tested here did work. So finally, we need to see whether we're hitting this also for the highest level of complexity, which is human. We don't work with human samples. So basically what we did is to team up with Edu Badle, researchers studying colorectal cancer in our institute. And basically what these guys want to do is to inhibit the Wnt pathway, but below the beta-catenin level, right? Because this is what achieves, at least genetically, the effect of blocking the progression of colorectal cancer stem cells. So these guys had information, again, gene expression on 10 different lines that were patient-derived, and they were achieving this through a genomic blockade. So CRISPR on these patient cancer cell lines, we had the information, they gave it to us, and our idea was to look for compounds able to mimic these genetic effects in here. So in this case, we used all the compounds in the broad collection, plus some compounds that we had in our in-house library. And at the end, we identified something like 3,000 different compounds that we thought might do the effect. We filtered them using different bioinformatics criteria that we had in the lab. And finally, we ended up with a list of 357 compounds that we thought might do this effect. So we tested them experimentally. Of course, this is bioinformatics, it's not a miracle, but just to give you an idea of the real results, this is unpublished yet. So for the 357 compounds that we identified computationally, we saw the desired effect in 90 of them. While if we randomly pick, according to chemical diversity, a similar number of compounds, this effect is achieved by only 4 compounds. So this means that by playing with these different signatures, etc., we are achieving a 23-fold enrichment in here. So this is cells, but these guys work with a human patient-derived organoid. So this is kind of the last level of complexity before going to humans. So we could not test, because of time and money reasons, the 90 positive hits. We selected, again, according to diversity, 11 of them. And so far, we've been able to validate 7 out of these 11 in patient-derived organoids, right? So it seems that somehow, so of course, the numbers are not perfect, but are much nicer than the one that I showed at the beginning in this drug discovery funnel that is tragic, right? You start from thousands of compounds and you end up with 1. So here you start with a couple of hundred, you end up with 90. So that's not bad. And basically what we have done here, what I presented, is this use of networks and network embedding techniques to develop these small molecule bioactivity and biological descriptors that are suitable for machine learning. Again, it's trying to condensate all the information that we have on biological entities, on a small molecule bioactivity in the form of vectors, because this is ideal for the computers. We can play with them. So we now provide these 1,000 context-dependent signatures to quantify the support and the novelty in any new experiment that you guys might do in the lab. And what we're doing is blending this type of small molecule and biological descriptors to kind of reintroduce the complexity in the first steps of drug discovery. So Sergio yesterday showed a similar picture in here. I'm going to develop it a little bit more. So basically, what we're doing here is analogous to the Indian blind man parable, basically. So I don't know if you're familiar with it, but basically it's a bunch of Indian blind men that they go into the jungle and they bumped into an elephant, right? So they're blind, so they don't know it's an elephant. So this one in here thinks that he's found a rope, and this one says, no, it's not a rope, it's a wall because I cannot pass. Or no, what you mean is a tree because I can hold a trunk. So the idea here is that each guy has partial information. So if they all are able to combine the partial information, maybe they can come up with a more faithful representation of the elephant, which is what they really hit. So same in here, by combining different types of descriptors of a small molecule bioactivity, different types of descriptors of the biological reality, maybe we can come up with a better idea of what it is that we need in the chemistry to really target the biological pathological mutations. As you can imagine, I did almost zero of this work apart from presenting it and getting some resources. So this is my team, a bunch of postdocs, a few PhD students and visiting students, and we're also open to recruit new motivated people, either from the chemistry side, from the biological side, or from the AI ML side. So if you want to come to sunny Barcelona, just drop me an email and I'll be happy to talk to you. And I'll use the last minute to advertise a conference that we're putting together, that we've put together for next year. This is in Barcelona, is myself and Trey Eidecker. So if I'm involved, Trey is involved, you can see that even if it's AI and drug discovery and biomedicine, there's going to be a lot of network biology in there as well. So this is the venue of the conference. So it's not just random pictures from Barcelona. So this is where the conference is going to take place, in the Hospital de Sant Pau, and it's completely free. So the conference is free. We give you lunch and dinner and breakfast, so you only have to take care of the accommodation. So the inscriptions are not open yet, but it's something that's going to happen in September, right after the summer break. And that's it. So thanks for your attention, and I'm happy to take questions.
Go ahead.
Gary.
Hey, great talk. It's great to see so much progress since the last time I heard about this. And one question I have is about images. Are you using images like, recursion, and other people are collecting huge amounts of images, and there's seems to be a rich amount of information about biological activity in them? I see you have some images. I'm just wondering if it's part of the plan.
So it's the answer is yes. In fact, one of the published bioactivity spaces is what we call cell morphology, is exactly the type of data that recursion and others are providing. We have others, so we work, this is the public part. We also work in collaboration with other pharma companies. So we have different bioactivity spaces done on proprietary data. So we have that. That's a short answer. A little bit more elaborated answer is that, at least, hope nobody from recursion is here, in our hands, it doesn't provide much information.
Okay, but I'm also wondering if the images that you have for the output are used to assess bioactivity, not just cell growth or something like that, but like more richer.
No. We take the measurements that they do.
Very, very exciting talk. I guess one thing I was wondering is, because of the number of different scales that you can operate on with a network like this, and the variety of examples that you showed, if your end goal is to find a useful therapeutic, how do you decide the appropriate entry point experimentally? So, you showed knockdown data, cell lines, mouse models.
So this is key. Great question. So basically, what we presented in the first version of the chemical checker were these 25 bioactivity spaces that they're trying to condensate all the information that we have in there. This is way too general for any problem, right? Mm-hmm. So just going back to the links 1000 example that somebody was mentioning before. So we're dealing here with 1000 different cell types. So if you're specifically interested in whatever melanoma, for instance, instead of these 1,000 cell lines, you can do exactly the same descriptors only on the 15 cell lines that have to do with the skin. And the same happens for the biological part. So the biotech is super general. This is what we wanted to present for the possibilities, but then for individual problems, you should be more specific, both on the bioactivity side. There's a protocol. I mean, you can do it yourself. It's under review, but the GitLab and everything is completely open, so you can create your own bioactivity spaces. But also at the biotech level, you should really control the type of metapath that you use and the type of information that goes into these descriptors. That's what I was saying, that we have these biotech supports. Out of the 1,000 context-dependent descriptors, you can pick the 4, 5, or 6 that are really more informative for your particular problem. Yeah, this is super general. For specific problems, you should be more specific. Sure.
Hi, nice presentation. Actually, I was quite excited about the example in the Alzheimer's disease context. And the reason why I'm excited is because, of course, it would suggest that actually by having this APP mouse model, you actually predicted the molecules that you could use to actually control the effect. The problem is that neurodegenerative disease have shown to fail when people are trying to target, let's say, the mouse model with APP. And actually now the community is moving around to try to figure out beyond APP. So basically we cannot model a complex disease with only one protein, right? So my question is now your system able to interrogate perhaps several thousands of genes that we don't understand what it's doing, but perhaps we could predict the effect?
Yeah, so I'm sorry, I didn't elaborate for the sake of time, but basically one of the models that we're using is the triple transgenic. So this means that it's not only APP, so it's APP, it's presenilin, and it's tau, right? So it's the 3 of them. Then the other thing, so this is one of the models that we're using. The other 2 is true, that is knock-ins that go directly to the APP. So the other thing that I didn't mention is that, as I said, when we play with mice, we cannot use any funny component that we have in the drawer. So we're using approved drugs. And basically what I didn't mention is that these 6 compounds that we found, we selected them because we saw, I mean, in fact, the computational system was giving us 80 different compounds. But we saw that many of them were clustering, were either NSAIDs, so non-steroidal anti-inflammatory drugs, or antihypertensives. So basically what we did was to pick 3 of each that nobody had related to Alzheimer's disease yet. And these are the 6 drugs that we tested, right? 4 of them did work. Then again, we're not a hospital, but we have colleagues in hospitals. So since these drugs are NSAIDs or antihypertensives, it means that a lot of people are taking them in a chronic manner. And basically what our colleagues have done is to run a retrospective analysis, and they see that indeed, so the elderly that are taking some of these drugs, the incidence of Alzheimer's disease is reduced. So these statistics doesn't prove anything. It's not prospective assay, but it seems that even at the highest level of complexity, this might be doing something.
Yeah, but again, coming to my question, instead of feeding 3 genes or so, could we give several hundreds?
Yeah, I mean, maybe I wasn't clear here. So we're not using 3 genes. So we're using full differential gene expression and proteomics of the hippocampus. So we're dealing with 20,000 genes. It's just that the animal models. So of course, these are not Alzheimer models. Are models for APP or triple transgenic. So they come from 3 individual mutations. But the input to our system is the result of the full transcriptomics and full proteomics of the hippocampus of these animals. I'm sorry. Yeah.
One short last question, please.
Sure. Yeah, I'm just curious. So, what are the databases you are using for the biological activity data for compound? And second question: so, were you incorporating the siRNA, RNA screening or RNAi screening in the process?
We are not in the chemical checker, because everything has to be related to small molecules there. But yes, in the biotech. So in the biotech, we have ways of targeting a gene that could be through small molecules or could be through interference or hairpins, etc. And then if we have a readout for this interference, either in gene expression or in cell viability, etc., this is incorporated in there. Yeah.
Thanks.
Sorry, I forgot to say that everything that we do is completely free and open source. So if you go to our GitLab, etc., you will find all the details of the different methods.
Can you please show the slide for the conference for next year?
Yep. It's not open yet, as I said. It will be open in September.
Everybody is taking the tickets now.
But not yet, September.
Great. Thank you.
Thank you.